Hello friends, today we are going to discuss a very common and well-known topic, cell signaling. What exactly this cell signaling means and where does it exist or occurs in environment or nature? See, in every living being, there is need of communicating with somebody. Even when cell wants to operate some functions on its own, or if we think of any developed higher living being, like say a human being or plant, then whatever we think, we need to communicate it to a required organ. For example, we think for movement of our hand. This is thinking operated in our brain, and then hand starts moving. So this is a kind of communication brain cells and muscle cells in the hands have. So cell signaling that way is very common thing. What exactly is cell signaling? It is a phenomenon of communication with each other through extracellular messenger molecules. Now there is one more word involved here, is extracellular messenger molecules. When say two individuals communicate amongst themselves, what do is we generate some kind of a signal. That signal can be in the form of a voice, signal can be in the form of our facial expression, signal can be in the form of any such communicative language. When a person who wants to communicate generates a signal, then that signal is transferred to the person who considered as a target person to whom that signal is to be received. Then that person receives that signal and then acts accordingly. So this kind of cell communication needs a messenger within those two individuals who are to communicate. When it is discussed at a cell level, that time this message is in the form of some chemical molecules which are actually coming out of a cell. So those message carrying things are known as extracellular messenger molecules. So what exactly is this cell signaling or cell communication? Is phenomenon of communicating with each other through extracellular messenger molecules. When say there is a cellular communication, then that cellular communication acquires much significance in the development of varieties of organisms. When say a newborn is being developed and from 2n fertilized cell, when it is to give you varieties of organs, cells from every organ then also have 2n number of cells, 2n number of chromosomes. Genetically all those cells are similar genotype wise, but then during differentiation, the process is known as differentiation. During differentiation, what happens is some kind of a signal is generated there, and those who are to receive that signal and to operate that signal, to respond to that signal, undergo some modifications, and then we can see there is development of different organs in the body. Even when one cell wants to communicate to the other, say for any particular purpose, that time also this kind of signaling mechanism is operated through extracellular messenger molecules. Basically, there are three main types of signaling mechanisms: autocrine signaling, paracrine signaling, and endocrine signaling. We are going to discuss each of them. Autocrine signaling, paracrine signaling, and endocrine signaling. All three of these mechanisms are operated through one of these three ways. It is via intercellular chemical messengers, means signal is generated from a producer, from a signaling cell, and through intercellular chemical messengers, these messages are operated, these signaling mechanisms are operated, or via gap junctions also they are operated, via cell surface proteins also they are operated. We will be seeing details of all these three also. First of all, we will see what is autocrine signaling. When auto is self, say, when one cell produces a signal, and that signal goes out of a cell and when comes back onto the surface of the same cell who is producer, and then that signal gives some impulse to the cell and then cell responds accordingly. That mechanism is known as autocrine signaling. If you see mechanism of interferon production, in response to some chemical stimuli, animal cells produce interferons. And interferon can also be a chemical messenger, but to behave as a chemical messenger, it should come out of a cell. When that interferon comes out of a cell, that time this interferon acts on the same cell who has produced it from outside. Actually, production is from inside, but interferon acts from outside. And then this cell produces two important substances in response to the stimulus from interferon, due to which the cell becomes antiviral then. The mechanism can be clarified here with this animation. You can see this is a cell which is producing signal. These yellow colored spheres are actually signaling molecules produced by this cell. They come out of a cell, then they function as extracellular messengers and they go back and act at the receptor sites of the same cell. Those receptors are for receiving the signal molecules. So this kind of mechanism is known as autocrine signaling mechanism. Then there is paracrine signaling mechanism. There is producer cell, signaling cell who produces signal molecule. That signal molecule comes out of a cell, but that signal molecule cannot go at a far distance in the body. That is because the signal may be unstable, is not very much stable, or may be degraded by enzymes within short period of a time. So within how much period that signal remains active, during that period only that signal keeps giving signals, signal molecule keeps giving signals, and that is how the cells which are at a close proximity to the producer only can get stimulated by these kinds of signals. So this mechanism is a paracrine signaling mechanism. In the animation you can see this is a cell who is producing signals, and these are the signal molecules produced by the cell, and these molecules keep acting onto the other cells, but those cells are not at much distance, they are nearby, present nearby to the producer cell. So this kind of mechanism is known as paracrine signaling mechanism. Then there is another mechanism, rather very significant and very prominent mechanism operated in most of the multicellular living beings. It is endocrine signaling mechanism in case of human beings, animals, and you can see this kind of mechanism is very much prominent. Endocrine signaling mechanism. See what this kind of mechanism, when we listen to any sound, say a very loud sound, then immediately our heart starts beating very fast. Function of listening to the sound is performed by our ear. It is actually internal ear. The waves hit the diaphragm in the middle ear, and then there are changes operated in the middle ear, and those changes then are percolated through internal ear to the brain, to the nervous system. So then that nervous system analyzes what kind of sound was that, and then gives some impulses. Those impulses are in the form of some signaling molecules, and those signaling molecules reach to the heart, and then heart starts beating very fast. Or when we see for a delicious dish, any delicious thing, we start salivating. Same way it is operated through eye. Eyes see that, or our nose when smells any particular delicious part. Nose works, nose takes the impulse, those impulses are sent to the nervous system. Nervous system generates a signal, means nerve cells generate a signal, and those signal molecules are then going towards salivary glands in the mouth, and then we start salivating. So this kind of mechanism needs travel of signal molecule at a distance from the nervous system up to heart muscles, say, from a nervous system up to salivary glands in the mouth, say, or elsewhere in the body also. So there is need of a travel of those molecules at a distance. How do these molecules travel? Actually, these molecules travel through some circulatory system, and the molecules are produced by some endocrine glands, secretory glands in the body. So these types of signals which are produced by signal producing cells and then are transferred through blood stream, through circulatory system to a target cell, and then target cells respond. So this kind of signaling molecule or this kind of signaling mechanism is known as endocrine signaling. In the animation you can see that this is a signal producing cell. Here you have one blood vessel shown, and this is a target cell. So when signaling cell keeps producing some signals, those signals are flowing through circulatory system, and signals are also can be percolated out of the blood vessels, and they reach to the target cells, and then these target cells respond. Follicle stimulating hormones, for example, they travel from the mammalian brain to ovary, where it triggers egg release. So see the distance. Signal is generated in the brain or in the follicle stimulating hormone, and then that hormone travels up to the egg release site where those ova are produced, and then those ova are released. So traveling of a signal at a distance is needed, and that is how it is known as endocrine signaling mechanism. Then we have seen there are three ways of operating those signaling mechanisms. One was via intercellular chemical messengers. Of course, every signal has to come out of a cell and then to act either on the same cell or on the nearby cells or at a distance. So when two different cells are communicating, then that signaling mechanism is known as intercellular signaling mechanism, or that way is known as intercellular chemical messenger. Cells send out signals in the form of specific chemical messengers that the target cell transmits into a biochemical reaction. What happens in the target cell then after receiving that signal is some biochemical changes are operated in the target cell, and that is how that cell responds. In the animation you can see this is where you have a signaling cell who is keeping producing signals, and those signals are reaching to the target cells. So this producer cell and the target cells are different. That's why this kind of signaling way is known as intercellular communication or via intercellular chemical messengers. Then second way of communication is via gap junctions. There are some cells which are connected to the other cells through some protein channel. In case of plant cells, this can be seen in the form of plasmodesmata. One cell is connected to the other cell through plasmodesmata. And when signals are produced by signaling cell, those signals, instead of coming out of a cell, signaling molecules go through that gap junction or through a channel present in between those two cells, protein channel present in between these two cells. And when those signaling molecules reach to the target cells, some biochemical events are operated in the target cell, and that is how that target cell undergoes some changes and shows a response. In animation you can see this. See, signaling cell is producing these signaling molecules. Signaling molecules are going through this gap junction, reaching to the target cell, and that is how this target cell is responding to the signal. So this is the way of communication via gap junctions. There is a third way also of communication, that is via cell surface proteins. We all know that immunological functions actually are through communications of immunocompetent cells. When an antigen is incorporated into the body, and any antigen processing cell processes that antigen inside and presents a required component of that antigen on the surface, then this antigen component presented on the surface of one cell, which can be considered as a T helper cell, or it can be a dendritic cell, or it can be any antigen processing cell. So when that processed substance is present on the surface of one cell, then that can be a signal for another cell. This can be a kind of antigen processing cell, and this can be a kind of target cell for whom this green colored substance is actually a signal substance, and this target cell also has receptor molecule on the surface for this signal substance. So when these two bind, it is actually a surface interaction in between these two cells. So communication of a signal from this cell through surface to this cell, and then showing response by this cell through some chemical messengers or through some biochemical reactions is seen. So this is because communication through contact of cell surfaces, this way of cell communication is known as cell communication via cell surface proteins. Okay, then the nature of signaling pathway. There are actually two parts of signaling pathway. One pathway is operated extracellularly. When a signal producing cell produces signal, releases it out, and then that signal is transferred up to the surface of another target cell. So this track from the producer up to the surface of target cell is known as intercellular signaling part. The first one is intercellular signaling part. Then when that signal is received on the surface of a cell, actually that signal is registered. The technical word is receiving signal at the surface on a target cell is registering signal on the surface of the cell. And then whatever changes are operated inside till that cell responds, till that cell shows a response in the form of some effectivity. Whatever happens now inside is in response to that signal, but because it is happening inside the cell, that part of signaling mechanism is known as intracellular signal transmission chain. So total signaling pathway is having two parts to be operated. One part is intercellular signaling part, which is operated outside the cell, and second part is intracellular signal transmission chain. This intracellular signal transmission chain also can be known as signal transduction. We'll be discussing it in a short while. So there are some steps involved in cell signaling. What are those steps or cell communications? The first step is creation of specific signals by a signaling cell. I have elaborated this point before also. Creation of a specific signals by a signaling cell. What happens when exactly are signals generated? There are some environmental influences cell always is influenced by. Some environmental conditions cell is always influenced by. That environmental condition can be a nutrient composition, presence or absence of, or a particular concentration of a nutrient. It can be, say, changes in the temperature. It can be changes in the pH value. It can be changes in the pressure, presence or absence of a salt, then concentration of salt. So many such things are there. Or listening to a loud voice, say for example for human being, listening to a loud voice, or for plants having sunlight, presence of sunlight, or absence of sunlight, or presence of CO2, or such chemicals presence or absence can be a impulse for creation of specific signals in the signaling cell. So this is the first step, creation of specific signals by signaling cell. Then second step is transportation of signals to the target cells. It means when signals are synthesized inside the cell, those signals are to be secreted out by a signal producing cell, and then these secreted signals are to be transported up to the target cell. Understood? So this can take place through gap junction or intracellular chemical messengers or through blood vascular systems. So that is how transportation of signals to target cells, or when for paracrine and endocrine signaling. For paracrine, why it is paracrine? Because signal is transported to a very short distance. So transportation of signal is another step. Then registering signals on the target cells. Target cells have particular receptors for signals on their surfaces. They are typical for specific signals. So there is a lock and key type of mechanism for a signal molecule and a receptor present on the cell surface. For example, when antibody production is a response expected from any B lymphocyte, there is a epitope present on the surface of an antigen, or maybe on the surface of antigen processing cell, processing and presenting cell. Then there is a correlation in between. Suppose this is a kind of antigen, then there has to be a typical receptor site to fit to the surface of an antigen on the surface of target cell. So this, when antigen or any signal molecule fits onto the receptor site properly, that is the mechanism known as registering the signal on the target cell. It is actually taking place at the surface of a cell. But registering doesn't mean simply binding of a signal molecule to the receptor. It is not necessarily only this part, because once this binding takes place, then the mechanism is operated inside the cell from the membrane part itself. So when mechanism starts getting operated, that is known as registering the signal inside the target cell. Then fourth step is transmission of signal into the target cell. Means when the signal mechanism or signal molecule is registered on the surface, then to show response by a target cell, there is need of operating some biochemical changes in the target cell. So that is known as transmission of signal into the target cell. Fifth step is processing of signals further with the help of intracellular signal chain. Actually, when any target cell is to respond to a signal at the surface, then so many changes are required to take place inside that target cell. Because say for example, when any target cell is to produce antibodies is a kind of a protein, unless that cell receives a signal from the surface, that cell doesn't produce antibodies. Does it mean that when cell is not receiving signal, that cell doesn't have genes for production of antibodies, production of those kinds of proteins? Genotypically, that cell is capable even when signal is present or signal is absent. Genetically, cell is capable. But only when cell receives the signal of antigen on the surface, then some biochemical changes are operated inside the cell, and those biochemical changes finally give a product which de-represses those genes which are expressed and produce typical type of proteins. Many times when our salivary system also, salivary glands also salivate. So secretion of saliva is a function operated. For that secretion part, some biochemical changes are required to take place in the cell which is secreting that saliva. So processing of signals further with the help of intracellular signaling chain is a fifth step. And sixth step is termination of a signal. Means when that cell responds properly, then there is termination of a signal molecule. So total signaling mechanism or a communication amongst cells, this mechanism is operated through these 6 steps: creation of specific signals, that is generation of specific signals by signaling cell, then transport of signal from a producer cell up to target cell, then reaction of signal on the surface of a cell, registering of the signal on the target cell, then transmission of this surface interaction inside the cell, then some biochemical changes are operated for actual production of the response and then showing the response, and finally termination of a signal. Now, I made mention about the signals in the first step, creation of specific signals. What exactly are these signals? These signals are nothing but some kinds of chemical messengers. These are some kinds of chemical species produced. Varieties of chemicals are seen produced. Some proteins are there. Some enzymes are there. Then, or some chemical mediators are there. For example, suppose if you see a cytotoxic T lymphocyte produces some chemicals, then those chemicals can attract macrophages. So macrophage is a target cell there. Or when interleukin substances are produced, then those interleukin substances also carry out some modifications in the target cells. Some hypersensitive reactions we see. So reddening of skin, reddening of eyes, shedding of tears. In suppose allergic rhinitis, we see these reactions. So production of signal is performed, and those signals are actually some kinds of chemical messengers. Understood? Now, first 3 steps, that is creation of specific signal, then transportation of signal to target cell and registering it on the surface. Up to this, everything is outside the target cell. Understood? It is in between the producer cell and the responding cell or on the target cell. So these 3 steps are making what is known as intercellular cell signaling part. At the beginning also previously we discussed that total signaling process takes place through 2 major steps, 2 major processes. One process is intercellular signaling and the second process is intracellular signaling. So if we see these 3 steps are intercellular cell signaling parts, and these 3 steps, step number 4, step number 5 and step number 6, means after registering signal on the surface, what happens is step 4, step 5 and step 6, whatever is happening is inside the cell. So that is also kind of signaling mechanism, that is also kind of signaling part, but that signaling part is known as intracellular cell signaling part. Okay. Now, exact mechanism of cell signaling is shown in this animation. This yellow colored cell is a signaling cell and this orange red colored cell is a target cell. And you can see this signaling cell is producing signals, those light green colored molecules which are seen, produced, synthesized and produced outside the cell, secreted outside the cell, are actually signal molecules released. Signal formation and release is shown over here. And one of the signal molecule is shown traveling up to this receptor site on the surface of this cell. So this is a target cell. This is transfer of a signal from here. Then at the surface, signal is detected or is registered, and then signal is seen transmitted inside. And some biochemical responses take place. So you can see this part of a process up to the registering of a signal, because is outside the target cell, is intercellular signaling part. Understood? And the whatever things are happening inside this target cell, that is signal transmission, then operating some biochemical changes here, and then biochemical response is shown by the cell, and finally we can see the effect at the target cell level, maybe production of some enzymes, maybe production of some proteins, maybe secretion of something, whatever, or blast transformation, increasing size of the cell, or getting attracted towards some part. So whatever changes are seen here in the target cell in response to those signals, all the changes to be operated are through whatever signal is transferred inside, whatever transduction takes place inside, whatever signal communication is done inside. That's why this part of signaling mechanism known as intracellular signaling mechanism, or is also known as signal transduction. You can see in animation that this signal is transmitted here on the surface, it is registered, and then whatever happening inside is a intracellular signaling transduction. Okay. Now, important mechanisms for activation of signaling proteins are: number 1, chemical modification of signaling proteins. Number 2 is triggering of enzymatic activity. These are some of the mechanisms which are need to be operated for activation of signaling proteins. Then third is triggering of conformational changes dependent upon, say, some chemical impulses, some environmental impulses or influences. There can be chemical modification in the signaling molecules, or some enzymatic activities also can be seen triggered, or some conformational changes also take place, triggering of conformational changes. Then increase of the concentration of signaling proteins and co-localization of signaling proteins. Actually, some of these mechanisms are operated inside the cell also. Means whenever signal is received or registered at the surface, then some proteins get activated, some proteins undergo conformational changes, some enzymes get activated, some enzymes act, they produce some biochemical changes. So these, or there is co-localization of signaling proteins also inside the cell. So these kinds of mechanisms are important for actually signaling process. This can be operated through proteins. Majority of the proteins are involved here. Now, the actual important part, the first part was intercellular signaling, where signal is produced by a signal producing cell, and that signal is transferred up to a target cell. What is happening inside the target cell is a major part, that is actually the important part, because what response we see is on the target cell. So that is intracellular signal transduction or intracellular signal communication. So what happens in this is, there is, what exactly is intracellular signal transduction is transmission of signal from the surface into target cell. Understood? We have seen. Suppose if you see this is one cell. Understood? And here on the surface, this cell has one kind of a receptor. And suppose this is going to be a signal molecule which can perfectly fit into this receptor molecule. When signal is registered here on the surface, then some part inside is operated. That beginning of operation of this part inside the cell begins from the membrane inside directly, and then some chemical changes take place, some modulators are produced, many proteins are involved, and finally the process may be operated at a DNA level. Suppose this is double stranded DNA, there are some genes which are to be de-repressed, and de-repression of the genes may result in production of some proteins. I told, suppose interferon does in this way, then it results in production of two important proteins. Those two important proteins, suppose this is one, this is another kind of a protein. There are genes for production of these proteins on the DNA molecule, but those genes are to be de-repressed, and this de-repression mechanism is begun from registering of the signal here at the surface. If suppose this is interferon, then when interferon binds to the receptor here at the surface, this production of these two proteins, which is actually 2'5' oligoadenylate synthase, and another one is protein kinase, due to presence of which this cell becomes antiviral. So this kind of mechanism when is operated inside the cell. So all those changes, whatever are operated inside the cell, are known as intracellular signal transduction. Now, second part here is some components are involved for this part. Whenever signal registered at the surface is to be operated up to effector level, then there are many components actually needed to be involved in the process. They can be proteins or many small molecular messengers are also there. Then the process is, incoming signal is passed from the receptor to the downstream proteins, which themselves have other proteins as the next partner in the signal cascade. Then there are some adapter proteins also involved. Means when one protein needs to communicate it to the next protein, some adapter proteins are there which are actually not necessarily enzymatic, and they can communicate the signal, they can pass that signal further. So those adapter proteins are also involved. And participating signal proteins can be enzymes, or they can act as connectors to recruit the other proteins in the signaling pathway. So these are adapter proteins. Now this is a diagram where I have shown that this is signal molecule, this is a membrane of the target cell, this is a receptor site, signal molecule is produced by some producer cell, and that is being transported here on the surface of this cell. This is the membrane of that cell, and this is the cytoplasmic portion or inside the cell portion. So the receptor, this is the receptor molecule. So if you look for this, there is E1, E2, E1 is an enzyme bound to the membrane, E2 also is an enzyme bound to the membrane. These are membrane bound components, and these components are known as sensor kinases. In most of the signaling mechanisms, there are some proteins which get phosphorylated, and for phosphorylation the reaction known as kinase reaction. So those proteins are known as sensor kinases, those enzymes are known as sensor kinases. So beginning of signal transduction from the surface is through sensor kinases, because those proteins are membrane bound. The process begins from those membrane bound proteins. Then through adapter proteins to non-membrane bound proteins, the signals can be transduced to enzyme 3. Now enzyme 3 can act on A, can convert it to B, and B can be additional intracellular messenger molecules which then keep acting on to other molecules. Maybe these enzyme molecules are made active, or some protein molecules are made active, and those protein molecules then can de-repress some genes present on the DNA molecule, and that is how that cell produces substances to show effect by the target cell. So these are the sensor kinases, and this is response regulator. Those proteins which are not bound to the membrane are response regulator, and those proteins which are bound to the membrane but can undergo phosphorylation are sensor kinases. So these two different groups of proteins are involved in these kinds of signal transduction. So these signal transduction mechanisms, or these kinds of signaling mechanisms, are known as two component regulatory or signaling systems, or two component regulatory mechanisms or signaling mechanisms. Now there are some examples of two component regulatory systems. Two component signaling system or two component regulatory system is very much ubiquitous in prokaryotes, but these are not the only sources to have these two component regulatory mechanisms existing. There are many more examples in case of plants as well as animals to show two component regulatory system. Particularly some simple examples we will discuss to understand what those two component regulatory systems are. Motility, for example, in case of bacterial cells or in prokaryotes, cells move from one place to the other. Now this movement of a cell from one place to the other is not a purposeless movement, in response to some chemical stimuli, in response to some physical factors like presence of oxygen or absence of oxygen or a gradient of oxygen, cells move. Suppose if cell is aerobic, means requiring oxygen for its multiplication, the cell can move from the region having less of oxygen toward the region of having more of oxygen. Or when there are chemicals, two types of chemicals can be there to respond. One is attractant. For example, for bacterial cells, glucose can be an attractant, or for a bacterial cell, phenol molecule, dettol molecule can be repellent. Understood? Antibiotic can be a repellent. So when cells respond to those attractants or repellents or to gradients of different environmental factors like temperature or oxygen level, then cells move. So this movement of a cell is an example of a two component regulatory system. It means cells actually respond to a signal, a chemical signal, and then cells move towards the attractant or they move away from a repellent. So this process of motility is an example of a two component regulatory system. Then nutrient uptake mechanism. Every time cell is not ready to uptake any nutrient. There are many nutrient molecules, there are many nutrient varieties which are inducible for the cell. Means, for example, lactose. Cell is not ready enzymatically every time for a lactose. So for utilization of a lactose, many such molecules are there, many such nutrients are there. So for nutrient uptake also, there is a two component regulatory system operated in some cells. Then for cell division, you see, cell division is a cell differentiation process. Every step is regulated. Where is exactly septum to be produced, how longer the elongation should be there in the cell from formation of a daughter cell onwards, then where exactly mesosomes are to be produced, how exactly DNA should be replicated. All those processes are time, place and quantity bound. So those cell division processes are also many times seen two component regulatory systems. Then bacterial virulence. When suppose in case of many experiments have been performed in case of Pseudomonas varieties. So when one cell, is entering into the body also doesn't keep quiet. Body also fights against. There are many defense mechanisms operated in the body itself. Suppose pathogen enters into the body, then body immediately acts at its first defense level, then acts at the second line defense level, then finally acts at a third line defense level. And when one pathogen is present in the body, that pathogen knows if there is no chance or there is no way out if I keep producing some toxins. So one cell doesn't produce. When number of cells are present inside the body, what is known as quorum, we'll be seeing that part in some other videos actually. So when quorum part is complete, then cells keep producing some substances which are involved in increasing virulence of the cell. So bacterial virulence is also a two-component regulatory system. Then what is exactly significance of cell signaling mechanism? Why nature has a cell signaling mechanism? Particularly if we think for higher living beings, multicellular living beings, anybody can understand that there is a obligatory need of cell signaling mechanism. Every part of body needs to be operated in a communication. Our brain cannot think separately, our hand cannot think separately, heart cannot think separately, lungs cannot think separately, we cannot walk separately, means legs cannot think separately. If every organ starts thinking separately, independently, then there cannot be a organized functioning of a body. So to have organized functioning of a body, definitely what is important is communication. Communication in between different organs, communication from nervous system to effector system, communication from say smell, means from nose through nervous system to effector system, communication through ear, means whatever sensory organs we have, those sensory organs sense the external impulse, and those external impulses then are processed and communicated to the effector part. So this is what is known as a signaling mechanism. When it is operated at a cell level, the mechanism is known as cell signaling. So that is how in case of eukaryotic system, cell signaling mechanism acquires very, very, very significant part. Some parts are mentioned in here. Multicellular living beings for total functioning of a body. Cell signaling mechanism is significant for total functioning of a body in case of multicellular living beings. Then cell differentiation process I told. Where exactly differentiation should take place? Suppose if a chick is being developed in an egg, 21 days is a gestation period for chick, for hen. So when 2N cell is developed in a egg and the 2N cell keeps getting differentiated, when and where exactly heart cells are to be produced, brain cells are to be produced, in how much quantity, where should be and how eyes should be produced. So all those mechanisms are operated through signaling. Some cells produce signals and some other cells respond to those signals. They get differentiated into eyes, they get differentiated into beak, they get differentiated into blood cells, they get differentiated into kidney, into liver, whatever things. So those are cell differentiation process. So cell signaling mechanism acquires very much significance in cell differentiation process. Then cell signaling mechanism is very significant in immune function also, because immune function is totally a cell communication function. One cell receives, because when suppose one say B lymphocyte produces antibodies, things doesn't over there. Even for production of antibodies by B lymphocyte, whatever mechanism operated inside is cell signaling mechanism. So immune function totally needs cell signaling mechanism. Then many organ functions also, our heart functions, liver functions, kidney functions, every part functions. How do they function? They function finally through cell signaling mechanism. Then many physiological processes, getting angry, becoming sad, or developing high fever, temperature, or whatever, suppose shedding tears, perspiration, means giving more of a sweat, all those things, physiological processes which are actually controlled through hormones. Hormones here actually, what hormones are finally? Hormones are a kind of signaling molecules, which actually act very much less concentration, and they can be dispersed at a very fast speed. You can imagine at what speed our heart starts beating after listening to a big or a loud sound. Immediately our heart starts beating. So how much time does it take to produce the signal, and how that signal is transduced, and how then the heart cells respond to that signal? So this takes place very fast. So its physiological processes operating into the body have a cell signaling mechanism. Cell signaling mechanism very significant and important for physiological processes. Prokaryotes are also not exception. In case of prokaryote system also you can see, because actually multicellular living beings we can understand there is need of communication from one organ to the other organ, or there is need of communication from one cell to the other cell, because every organ's function is different, and to coordinate those functions there is definitely requirement of a cell communication. So we can do understand the significance of cell signaling mechanism in higher living beings, in multicellular living beings. But prokaryotes like bacterial cells, do they need to communicate then? Yes, they do need. In spite of they being unicellular, for better fitment to the environment, for better suiting to the environment, for better suitability for their existence and for growth, they need to have a community life. And they do have. They do have. This is a separate branch for study in case of bacterial cells, in bacteria, that is known as quorum sensing part. Means cells keep communicating amongst group of the cells. Even though single cell can survive, single cell can lead a life, single cell can grow, that is how it is a unicellular life. Prokaryotes are unicellular living beings. They need to have, for better life, a community life. What is actually a requiring signaling mechanism, cell communication mechanism. So that is what is a quorum sensing mechanism. To get set into better manner to the environment, there is a requirement of cell communication. For example, say a luminescent cell is to luminous, cell is to show that luminescence. So there is need of a cell communication. There is need of a quorum sensing. Or when say there are F minus cells, female cells, and a male cell, that is F male, F plus cell, wants to meet to female cell, then there is some response expected from F plus cell, from a male cell. So this response is in connection to the signal produced. So these kinds of signal mechanisms are operated in prokaryotes also. So prokaryotes, in spite of being unicellular, to have a community life, they need to have a community life. They need to have a cell communication mechanism, cell signaling mechanism. That can be a separate video for quorum sensing. We'll be discussing for this in a separate video also. So these are the significant points for cell signaling mechanism. Now thank you all. Actually, if you have liked this video, then please subscribe my video. I will be putting some additional videos on YouTube. This video also is on YouTube. Please subscribe my YouTube channel. My YouTube channel is Nasir Shaikh, named by Nasir Shaikh. So please subscribe my YouTube channel to see subsequent videos. Thank you one and all again.